Cardiovascular system. The cardiovascular system consists of three interrelated components: blood, the heart, and the blood vessels. The branch of science concerned with the study of blood-forming tissues and the disorders associated with them is called hematology. Human has closed blood vascular system. In this system, blood is pumped through a closed system of heart, arteries, veins, and capillaries without forming indirect contact of blood tissues or blood cavity. Blood flows at high pressure. Physical characteristics of blood. Blood is slightly alkaline, pH ranging from 7.35 to 7.45. Blood constitutes about 20% of extracellular fluid, amounting to 8% of total blood mass. Total body mass. The blood volume is 5 to 6 liters in an average sized adult male and 4 to 5 liters in an average sized adult female. Functions of blood: transportation of oxygen, carbon dioxide, nutrients, hormones, heat, and wastes. Regulation of pH, body temperature, and water content of cells. Protection against blood loss through clotting and against diseases through phagocytic activity with the blood cells and antibodies. Components of blood. Blood is a connective tissue composed of liquid extracellular matrix called blood plasma that dissolves and suspends various cells and cell fragments. So, blood has two components: blood plasma, a watery liquid extracellular matrix that contains dissolved substances and formed elements which are called cell fragments. Blood is about 45% formed elements and 55% blood plasma. When the formed elements are removed from the blood, a straw-colored liquid called blood plasma or simply plasma is left. The separation can be either achieved by just leaving the blood undisturbed for some time, leading to settling down of the cells by centrifuging them. Then blood plasma is about 91.5% water and 8.5% solutes, most of which 7% by weight are proteins. Some of the proteins in blood plasma are also found elsewhere in the body, but those confined to blood are called plasma proteins. Hepatocytes synthesize most of the plasma proteins. This include the albumins, 54%, plasma proteins, globulins about 38%, fibrinogen 7%. Albumin, globulin, fibrinogen. If blood is allowed to clot, then the clotting factor present in the plasma are used up and hence the fluid left behind is called serum. Formed elements. The formed elements of the blood include three principal components: red blood cells, white blood cells, and platelets. Red blood cells and white blood cells are whole cells, platelets are cell fragments. RBC and platelets have just a few rules, but WBC have a number of specialized functions. Several distinct types of WBCs present in blood are neutrophils, lymphocytes, monocytes, eosinophils, and basophils. The percentage of total blood volume occupied by RBC is called hematocrit or packed cell volume. A hematocrit of 40 indicates that 40% of volume of blood is composed of RBC. The normal range of hematocrit for adult females is 38 to 46%, and for adult males it is 40 to 54%. In polycythemia, the percentage of RBC is abnormally high and the hematocrit may be 65% or higher. This increases the viscosity of blood, which increases the resistance to flow. Formation of blood cells. The process by which the formed elements of blood develop is called hemopoiesis or hematopoiesis. Before birth, hemopoiesis first occurs in the yolk sac of an embryo and later in the liver, spleen, thymus, and lymph nodes of a fetus. Red bone marrow becomes the primary site of hemopoiesis in the last 3 months before birth and continues as the main source of blood cells after birth and throughout life. Red bone marrow is highly vascularized connective tissue located in the microscopic spaces between trabeculae or of spongy bone tissue. In newborns, all bone marrow is red, but it is active in blood cell production. As an individual grows, as in adulthood, the rate of blood cell formation decreases. The red bone marrow and the medullary marrow cavity of long bones become inactive, and this is replaced by yellow bone marrow, which is largely fat cells. Red blood cells or RBC or erythrocytes contain the oxygen-containing protein hemoglobin, which is a pigment that gives whole blood its red color. A healthy adult male has about 5.4 million red blood cells per microliter of blood, and a healthy adult female has about 4.8 million. The cytosol of RBCs contain hemoglobin molecules. These important molecules are synthesized before loss of nucleus during RBC production and constitute about 33% of the cell's weight. Red blood cells are highly specialized for their oxygen transport function. Because mature RBCs have no nucleus, all their internal spaces is available for oxygen transport. RBCs have a biconcave shape which offers at least these advantages. If surrounded by a hypotonic solution, the RBCs can swell considerably before bursting. The biconcave shape is best suited for binding along the middle and this shape has large surface to volume ratio which increase the efficiency of oxygen transfer across the RBC membrane. And each RBC contains about 280 million hemoglobin molecules. A hemoglobin molecule consists of a protein called globin composed of 4 polypeptide chains, 2 alpha and 2 beta chains. A ring-like non-protein pigment called heme is bound to each of the 4 chains. Hemoglobin, hemocyanin, chlorochronin, hemoerythrin and pinaglobin. Few mollusks. Red life cycle. Red blood cells live only about 120 days because of the wear and tear in their plasma membrane undergo as they squeeze through blood capillaries. Ruptured red blood cells are removed from circulation and destroyed by fixed phagocytic macrophages in the spleen and liver, and the breakdown products are recycled. This is brought about the macrophages of the reticuloendothelial system. White blood cells, types of WBC, unlike red blood cells, white blood cells or leukocytes have nuclei and do not contain hemoglobin. WBC are classified as either granular or agranular depending on whether they contain conspicuous chemical filled cytoplasmic granules or vesicles that are made visible by staining. Granular leukocytes include neutrophil, eosinophil, and basophil. Granular leukocytes include lymphocytes and monocytes. Monocytes and granular leukocytes develop from a myeloid stem cell, and lymphocytes develop from a lymphoid stem cell. Then granular leukocytes. Eosinophil contains eosinophil granules and stain red-orange with acidic dyes. Basophil contains large basophilic granules and stain blue-purple with basic dyes. The granules of a neutrophil are smaller, evenly distributed and pale yak in color. The nucleus has 2 to 5 lobes as the cell age. The number of nuclear lobes increase because older neutrophils have several different shaped nuclear lobes. They are called polymorphonuclear leukocytes. Then agranular leukocytes. Even though so-called agranular leukocytes possess cytoplasmic granules, the granules are not visible under a light microscope because of their small size and poor staining qualities. The nucleus of a lymphocyte is round and stains darkly. Then monocytes are 11 to 20 micrometer in diameter. The nucleus of a monocyte is usually kidney shaped or horseshoe shaped, and the cytoplasm is blue-gray. The color and appearance are due to very fine xerophilic granules, which are lysosomes. The blood transports monocytes from the blood into the tissues where they enlarge and differentiate into macrophages. Then functions of WBC. In a healthy body, some WBCs, especially lymphocytes, can live for several months or years, but most live only a few days. WBCs are far less numerous than red blood cells, at about 5,000 to 10,000 cells per microliter of blood. Leukocytosis, an increase in the number of WBC about 10,000 microliter is a normal protective response to stress such as invading microbes, strenuous exercise, anesthesia, and surgery. An abnormally low level of white blood cells below 500 per microliter is termed leukopenia. Platelets. Besides the immature cell types that develop into leukocytes and erythrocytes, hemopoietic stem cells also differentiate into cells that produce platelets. Under the influence of the hormone thrombopoietin, myeloid stem cells develop into megakaryocyte colony forming cells that in turn develop into precursor cells called megakaryoplasts, which transform into megakaryocytes. Cells that splinter into 2000 or 3000 fragments. Each fragment enclosed by a piece of plasma membrane is a platelet thrombocyte. Platelets break off from the megakaryocytes in red bone marrow and then enter the blood circulation. Between 1,50,000 and 4 lakh platelets are present in each microliter of blood. This is disc-shaped, 2 to 4 micrometer in diameter, and has many vesicles but no nucleus. Platelets help stop blood loss from damaged blood vessels by forming a platelet plug. Their granules also contain chemicals that, once released, promote blood clotting. Platelets have a short lifespan, normally just 5 to 9 days. Aged and dead platelets are removed by fixed macrophages in the spleen and liver. Then blood clotting. Normally, blood remains in its liquid form as long as it stays within its vessels. If it is drawn from the body, it thickens and forms a gel. Eventually, the gel separates from the liquid. The straw-colored liquid called serum is simply blood plasma minus the clotting proteins. The gel is called a clot. So it consists of a network of insoluble protein fibers called fibrin, in which the formed elements of blood are trapped. The process of gel formation, called clotting or coagulation, is a series of chemical reactions that culminates in the formation of fibrin threads. Blood clotting involves several substances known as clotting factors. These factors include calcium ions. Several inactive enzymes are synthesized by hepatocytes, liver cells, and released into the bloodstream, and various molecules associated with the platelets are released by damaged tissues. The fundamental reaction in the clotting of blood is conversion of soluble plasma protein fibrinogen to insoluble fibrin. Following flow chart describes the process of blood clotting. Injured tissue, thromboplastin, blood platelets, plasma thromboplastin combines to form in the presence of Ca2+, prothrombinase, prothrombin, thrombin, fibrinogen, soluble, fibrin, insoluble process of blood clotting. And vitamin K is essential for normal blood clotting because it is required for the synthesis of prothrombin. Anticoagulant action of coumarin group of compound dicumarole is due to homologous structure to vitamin K, and hence they inhibit clotting. And anemia. Anemia is a condition in which oxygen carrying capacity of blood is reduced. All of the many types of anemia is characterized by reduced number of RBCs or decreased amount of hemoglobin in the blood. Among the most important types of anemia are following: inadequate absorption of iron, excessive loss of iron, increased iron requirement, or insufficient intake of iron causes iron deficiency anemia, in the most common type of anemia. Inadequate intake of vitamin B12 or folic acid causes megaloblastic anemia, in which red bone marrow produces large abnormal red blood cells. Insufficient hemopoiesis resulting from an inability of the stomach to produce intrinsic factor, which is needed for absorption of vitamin B12 in small intestine, causes pernicious anemia. Excessive loss of RBC through bleeding resulting from large wound stomach ulcers or especially heavy menstruation leads to hemorrhagic anemia. Megaloblastic anemia, pernicious anemia, hemorrhagic anemia. RBC plasma membranes rupture prematurely in hemolytic anemia. Then deficient synthesis of hemoglobin occurs in thalassemia, a group of hereditary hemolytic anemia. Destruction of red bone marrow results in aplasia anemia. Then heart, anatomy of the heart. The heart is located in the mediastinum. About 2/3 of its mass is to the left of the midline. The heart is shaped like a cone lying on its side. Its apex is the pointed inferior part, where its base is the broad superior part. The pericardium is the membrane that surrounds and protects the heart. It consists of an outer fibrous pericardium composed of tough, inelastic, dense, and irregular connective tissue, and an inner serous pericardium forms a double layer around the heart, outer parietal and an inner visceral layer. Between the parietal and visceral layers of serous pericardium is the pericardial cavity, a potential space filled with a few milliliters of pericardial fluid that reduces friction between the two membranes. Then layers of heart wall. 3 layers make up the heart of the wall. Epicardium, visceral layer of the serous pericardium, myocardium and endocardium. The epicardium consists of mesothelium and connective tissue. And myocardium is composed of cardiac muscle tissue, and endocardium consists of endothelium and connective tissue. Then chambers of heart. The heart chambers include two superior chambers, the right and left atria, and two inferior chambers, the left and right ventricle. The right atrium receives blood from the superior vena cava, inferior vena cava, and coronary sinus. It is separated from the left atrium by the interatrial septum, which contains the fossa ovalis. So blood exits in the right atrium through the tricuspid valve. The right ventricle receives blood from right atrium. It is separated from the left ventricle by an intraventricular septum and pumps blood through the pulmonary valve into the pulmonary trunk. Oxygenated blood enters the left atrium from the pulmonary veins and exits through the bicuspid or mitral valve. The left ventricle pumps oxygenated blood to the aortic valve into the aorta. The thickness of the myocardium of the four chambers varies according to the chamber's function. The left ventricle, which is the highest workload, has the thickest wall. Then heart valves and circulation of blood. As each chamber of the heart contracts, it pushes a volume of blood into ventricle or out of the heart into an artery. Valves open and close in response to pressure changes as the heart contracts and relaxes. Heart valves prevent backflow of blood within the heart. The atrioventricular valves, which lie between atria and ventricles, are the tricuspid valve on the right side of the heart and the bicuspid mitral valve on the left. The semilunar valves are the aortic valve at the entrance of the aorta and the pulmonary valve at the entrance of the pulmonary trunk. Here you could see a picture which depicts all this. Anterior view of frontal section showing internal anatomy of heart. Inferior vena cava present. Left common carotid artery. Left common carotid artery, left subclavian artery, brachiocephalic artery, aorta, superior vena cava, aortic valve, pulmonary valve, pulmonary artery, pulmonary vein, mitral valve, tricuspid valve, then inferior vena cava. So systemic and pulmonary circulation. The heart is actually two separate pumps. The left side pumps blood to the body, systemic circulation, and the right side pumps blood to the lungs, pulmonary circulation. Each side has an atrium and a ventricle. The left side of the heart is the pump for the systemic circulation. The circulation of blood throughout the body except for the air of the lungs. The left ventricle ejects blood into the aorta and blood then flows into systemic arteries, arterioles, capillaries, venules and veins, which carry it back to the right atrium. So yeah, we could see how this process is happening. Systemic and pulmonary circulation. First, left ventricle, aorta and systemic. In systemic capillaries, blood loses O2 and gains CO2, superior vena cava, inferior vena cava and coronary sinus. Then right atrium, deoxygenated blood, right ventricle through tricuspid valve, pulmonary trunk and pulmonary arteries through pulmonary valve. Then the pulmonary capillaries, blood loses CO2 and gains O2. The right side of the heart is the pump for pulmonary circulation. The right side of the heart is the pump for pulmonary circulation, the circulation of blood through the lungs. The right ventricle ejects blood into the pulmonary trunk and blood then flows into pulmonary arteries, pulmonary capillaries and pulmonary veins and carry it back to the left atrium. Cardiac muscle tissue and cardiac conduction system. Cardiac muscle fibers usually contain a single Cardiac muscle fibers usually contain a single centrally located nucleus. Compared to skeletal muscle fibers, cardiac muscle fibers have more and large mitochondria, slightly smaller sarcoplasmic reticulum and wider transverse nucleus. Once again, cardiac muscle fibers usually contain a single centrally located nucleus. Compared to skeletal muscle fibers, cardiac muscle fibers have more and larger mitochondria, slightly smaller sarcoplasmic reticulum, and wider transverse tubules, which are located at Z discs. Cardiac muscle fibers are connected via end-to-end intercalated discs. Desmosome in the disc provide strength, and gap junctions allow muscle action potentials to conduct from one muscle fiber to its neighbors. Some specialized cardiac muscle fibers are called autorhythmic fibers because they are self-excitable. Autorhythmic fibers repeatedly generate action potentials that trigger heart contractions. These rare fibers have two important functions. They act as pacemaker, setting the rhythm of electrical excitation that causes contraction of the heart. They form the conduction system, a network of specialized cardiac muscle fibers that provide a path for each cycle of cardiac excitation to progress through the heart. Components of the conduction system are the sinoatrial node or SA node pacemaker, the atrioventricular AV node, atrioventricular AV bundle of His, then bundle branches and Purkinje fibers. Electrical stimulation. The heart does not require outside stimulation. The sinoatrial node is a bit of nerve tissue that serves as a cardiac pacemaker. Stimulation from this node causes both of the atria to contract at the same time because the muscle tissue conducts the stimulation rapidly. The contraction doesn't spread to the ventricles because the atria and ventricles are separated by connective tissue. As a wave of stimulation or depolarization spreads across the atria, resulting in their contraction, another bit of nerve tissue called the atrioventricular node also becomes stimulated or depolarized. It conducts the action potential slowly to the ventricles. The slow speed is due to small diameter of the neurons within the node. The slow speed of conduction within the AV node ensures that the ventricles contract Stimulation of this node causes both of the atria to contract at the same time because the muscle tissue conducts stimulation rapidly. The contraction doesn't spread to the ventricles because atria and ventricles are separated by connective tissue. As a wave of stimulation or depolarization spreads across the atria, resulting in their contraction, another bit of nerve tissue called the atrioventricular node also becomes stimulated or depolarized. It conducts the action potential slowly to the ventricles. The slow speed is due to the small diameter of the neurons within the node. The slow speed of conduction within the AV node ensures that the ventricles contract after the atria contract. A bundle of His then transmits the impulse rapidly from the AV node to the ventricles. Then heartbeat. This is a spontaneous rhythmic contraction and relaxation of the heart to pump out and receive blood to form the body, to and from the body. Heartbeat are of two types, neurogenic and myogenic. On the basis of this, heart is differentiated into two types, neurogenic and myogenic. Regulation of heartbeat. Heartbeats at a fixed rate due to inherent property of the fibers of SA node, but it can also be controlled by nerves and hormonal mechanisms. Nerve regulation. Center of heartbeat regulation in heart. Brain is medulla oblongata. Sympathetic nerve system having accelerator nerves increase the heartbeat by secreting adrenaline hormone, whereas parasympathetic nerve system supplying it vagus nerve decrease the heartbeat by secreting acetylcholine. Then hormonal regulation. Thyroxine secreted by thyroid gland increases heartbeat indirectly by increasing basal metabolic rate, which requires more oxygen. Epinephrine and norepinephrine secreted by adrenal medulla also have a role of regulation. Epinephrine increases heart rate in emergency, while the norepinephrine increase heartbeat under normal conditions. Cardiac cycle. The cycle of events that occur in a single heartbeat is called cardiac cycle. It involves repeated contraction and relaxation of the heart muscle. Contraction when blood is ejected from the heart is called systole, and relaxation when the chambers of the heart are filled with blood is called diastole. The heart is said to be in a state of joined diastole when all its four chambers are in relaxed state. The cardiac cycle takes about 0.8 seconds and it is completed in the following phases. Isometric relaxation. Isometric relaxation is the first phase when all the valves are closed and the aorta and ventricles are relaxed. Isometric relaxation. Then atrial diastole. Both atria are filled with blood, having deoxygenated in right and oxygenated in left, and the atria and ventricles are both relaxed. At first, the bicuspid and tricuspid valves are closed, but as the atria gets filled with blood, their increased pressure eventually becomes greater than that in the relaxed ventricles, and the AV valves are pushed open. The first bicuspid and tricuspid valves are closed, but as the atria gets filled with blood, their increased blood pressure eventually becomes greater than that in the relaxed ventricles, and AV bundles are pushed open. The SL semilunar valves remain closed. This phase is also known as rapid filling phase. The blood flow into the heart then ceases as the pressure in the heart equals to that in the veins supplying it. Then there is a brief period of diastasis when there is no movement of blood at all. At the end of the phase, the two atria contract simultaneously by SA node and results in the blood being jumped into ventricles. This is called atrial systole. Ventricular systole. When the contraction of ventricles occurs immediately after atrial systole, the pressure in the ventricles rises, closing the atrioventricular valve, preventing blood from returning to the atria. When the pressure forces to open the semilunar valves of aorta and pulmonary artery to make entry of the blood into these vessels. The closing of atrioventricular valves during ventricular systole produces the first heart sound lub, ventricular diastole. During this, the semilunar valves are closed because of the blood forced back into the ventricles due to high pressure developed in the vessels. This causes the second heart sound dub. So, aortic and pulmonic valves opening, atrioventricular valves closing, ventricles contracting. Aortic and pulmonic valves closing, atrioventricular valves opening, ventricles relaxing. First heart sound lub, second heart sound dub. First heart sound lub means aortic and pulmonary valves opening, atrioventricular valves closing, ventricles contracting. Then blood pressure. It is the arterial pressure of blood exerted on the walls of arteries with each heartbeat. The blood pressure can be measured by sphygmomanometer. You might have seen that apparatus. Then blood pressure is expressed as systolic pressure in mmHg by diastolic pressure. So normal blood pressure has high systolic value, systolic on the top, and low diastolic value. Systolic pressure is higher because of increased pressure in arteries because of a contraction of heart and subsequent flow of blood in the artery at a pressure. Then cardiac output. The amount of blood flow from the heart over a given period of time is known as cardiac output. Cardiac output is equal to stroke volume of blood pumped by heartbeat into heart rate, ventricular systole per minute. Double circulation is the passage of blood twice in the heart through separate pathways for completing one cycle. Coronary circulation. Coronary arteries supply the heart muscles with blood. They have a very small diameter and may become blocked, producing a heart attack. Then blood pressure. The units of measurement of milliliters of mercury, mmHg. For example, 120 mmHg per 80 mmHg is considered to be normal blood pressure. The top number is referred to as the systolic pressure and the bottom number is the diastolic pressure. Blood vessels. Arteries carry blood away from the heart. Arteries have a thick elastic layer to allow stretching and absorb pressure. The wall stretches and recoils in response to pumping. These peaks in pressure are absorbed. The arteries contain pressure in the circulatory system, much like a balloon maintains pressure on the air within it. The arteries therefore act as pressure reservoirs by remaining or storing pressure. The elastic layer is surrounded by circular muscle to control the diameter and thus the rate of blood flow. An outer layer of connective tissue provides strength. Arteries divide to form finer branches or arterioles as they reach the destination. Veins. A vein starts by the union of venous capillaries leading to the formation of venules which then join and forms a vein. Venules drain blood from capillaries into veins. Veins are thin walls and large lumen and causes less resistance to blood flow. The blood pressure in the veins is low, so valves in veins help prevent the backflow. Then capillaries. The smallest blood vessels are capillaries. They are typically less than 1 mm long. The diameter is so small that red blood cells travel single file. Not all of the capillary beds are open at one time because all of them would hold 1.54 times the total blood volume of all blood in the body. The total cross-sectional area of the capillaries is greater than that in the arteries or veins, so the rate of blood flow velocity is lowest in capillaries. Blood pressure is highest in the arteries, but it is considerably reduced as it flows through the capillaries. It is lowest in the veins. Then interstitial fluid. The exchange substance between blood and the blood cells occurs in the capillaries. Capillaries are specialized for exchange of substances in the interstitial fluid. Nozzle in the body is more than 100 micrometers away from a capillary. This the thickness of 4 sheets of paper. Interstitial fluid surrounds and weights the cells. The fluid is continuously being replaced by fresh fluid from blood in the circulatory system. Blood cells take up nutrients from the interstitial fluid and empty wastes into it. By maintaining a constant pH and ionic concentration of the body, blood, the pH and ionic concentration of the interstitial fluid is also stabilized. Then circulatory routes. Blood vessels are organized into circulatory routes that carry blood to specific organs in the body. The two main circulatory routes are systemic and pulmonary circulations. Systemic and pulmonary circulations differ in two important ways. First, blood in the pulmonary circulation need not be pumped as far as blood in the systemic circulation. Second, compared to systemic arteries, pulmonary arteries have pulmonary and systemic circulations. The systemic and pulmonary circulations differ in two important ways. First, blood in the pulmonary circulation need not be pumped as far as blood in the systemic circulation. Second, compared to systemic arteries, pulmonary arteries have large diameters, thinner walls, and less elastic tissue. As a result, the resistance to pulmonary blood flow is very low. The systemic circulation includes the arteries and arterioles that carry oxygenated blood from the left ventricles to systemic capillaries, plus the veins and venules that return deoxygenated blood to the right atrium. Along the subdivisions of systemic circulation are the coronary circulation and the hepatic portal circulation. The pulmonary circulation takes deoxygenated blood from right ventricle to the alveoli within the lungs and return oxygenated blood from the alveoli to the left atrium. So left pulmonary artery to air sacs of right lung, right pulmonary veins, left atrium, then pulmonary trunk, right ventricle, left pulmonary artery, air sacs, alveoli of left lung. Next, hepatic portal circulation. The hepatic portal circulation carries venous blood from the gastrointestinal organs and spleen to the liver. A vein that carries blood from one capillary network to another one is called a portal vein. The hepatic portal vein receives blood from capillaries of gastrointestinal organs and the spleen and delivers it into sinusoids of the liver. Heart, inferior vena cava. 1 minute. Scheme of principal blood vessels of hepatic portal circulation and artery supply and venous drainage of liver. First, superior mesenteric vein, splenic vein, hepatic portal vein, liver, hepatic veins, inferior vena cava, heart, abdominal aorta, proper hepatic artery. Next, the lymphatic system. The cardiovascular system is considered a closed system because all of its tubes are connected with one another and none is simply open-ended. In other sense, however, the system is open to diffusion through the walls of the capillaries. This process is a necessary part of the functioning of the circulatory system, but it poses difficulty in maintaining the integrity of that system. Difficulties arise because diffusion from the capillaries is accompanied by the loss of large quantities of liquid from the cardiovascular system. When blood passes through the capillaries, it loses more water to the body than it reabsorbs from it. In a human being, about 3 liters of fluid leave the cardiovascular system in this way each day, a quantity amounting to more than half the body's total supply of about 5.6 liters of blood. To counteract the effects of this process, the blood uses a second open circulatory system called the hepatic system. Lymphatic system. The elements of the lymphatic system gather liquid from the body and return it into cardiovascular circulation. Open-ended lymph capillaries gather up fluids and carry them through a series of progressively larger vessels to two large lymphatic vessels which resemble veins. The lymphatic vessels contain a series of one-way valves like those of veins which permit movement in only one direction. These two lymphatic vessels drain into veins through one-way valves. No heart pumps fluid through the lymphatic system. Instead, like veins, fluid is driven through the lymphatic vessels when these vessels are squeezed by the movement of the body's muscles. Then lymph. It is an interstitial fluid or tissue fluid present between the cells of a tissue. This fluid is collected in tiny tubes called lymph vessels or lymph capillaries or lymph nodes, and fluid is now called lymph. Its composition is just like plasma except it lacks RBCs and large plasma proteins. It is colorless due to absence of hemoglobin. It transfers materials from the blood to the body cells and vice versa, therefore acts as a middleman. Here we could see a picture. Systemic circulation and pulmonary circulation, lymph node, lymphatic duct, subclavian vein, lymphatic vessel, valve, lymph node, lymphatic capillaries, systemic blood capillaries, arteries, pulmonary blood capillaries, and lymphatic capillaries. Some other terms like basal metabolic rate, not related to heart, but still we are going to learn this. The rate at which energy is used in performing metabolic reactions is known as metabolic rate. The metabolic rate measured under standard conditions with body in quiet, resting and fasting conditions is known as basal metabolic rate or BMR. BMR is generally determined by measuring amount of oxygen used per kilocalorie of food metabolized. Normally, BMR is about 24 kilocalorie per gram of body mass in adult males and 22 kilocalorie calorie per kilogram of body mass in adult female. This energy is sufficient for functioning of vital organs such as heart, lungs, brain, liver, kidneys, etc. in resting body. Extra calories is required for daily activities such as digestion, walking, etc. BMR decreases with age and loss of lean body mass. Other functions including environmental temperature, consumed food and beverages, stress levels, etc. also affect BMR. The primary organ regulating BMR is hypothalamus. Hypothalamus regulates autonomic nervous system, body temperature, food intake, feelings of rage and aggression. All these functions taken together regulate BMR. About 70% of energy expenditure comes from BMR, 20% of physical activity, and about 10% from thermogenesis or digestion of food. Then thermoregulation. Thermoregulation is the ability of an organism to keep its body temperature constant even when variation occurs in surrounding temperature. Thermoregulation is an important aspect of homeostasis, a dynamic state of equilibrium between an animal's internal and external environment. Organisms that thermoregulate keep their core body temperature within certain limits are also known as warm-bodied animals. Thermoconformists or cold-blooded animals, on the other hand, change their body temperature with a change in temperature outside the body. Almost all birds and mammals have a high body temperature, almost constant and independent of the surrounding air. This is called homothermy. Almost all other animals display variation in body temperature dependent on their surroundings. In humans, skin and hypothalamus gland are two important organs involved in thermoregulation. Normally, human body temperature is 37 degrees Celsius or 98.6 degrees Fahrenheit. Thermoreceptors in the skin send signals to thermoregulatory centers of hypothalamus that performs thermoregulation with the help of pituitary gland. At high temperatures, sweat glands under the skin are stimulated to secrete sweat, which causes high heat loss by evaporation. Vasodilation of arterioles allow increased blood flow through the artery, causing increase in heat loss by radiation and conduction. Metabolic rate decrease. The hairs in the skin lie flat, preventing heat from being trapped by the layer of still air between the hairs. This is caused by relaxation of tiny muscles under the surface of skin called the erector pili muscles at high temperatures. Then in cold temperatures, sweat production stops. Contraction of erector pili muscles lifts the hair follicle upright. This makes an insulating layer over the skin that traps heat. Then vasoconstriction of the arterioles occurs in superficial capillaries under the surface of skin, and the blood is flowed towards the warmer core of the body. This prevents heat loss from blood to surroundings and also prevents further drop in core temperature. Shivering is stimulated by the thermoregulatory center in the hypothalamus. This increases heat production through respiration and muscle cells. Nerve impulses stimulate the arterial medulla to release epinephrine and norepinephrine into the blood. This hormone increases cellular metabolism and thus heat production. Increase in levels of thyroid hormones in blood slowly increase the metabolic rate and thus body temperature. Then physical and chemical neural regulation of body temperature. A diurnal variation in body temperature is observed in both humans and monkeys, dependent on the periods of rest and activity, being lowest at 11 p.m. to 3 p.m. and peaking at 10 a.m. to 6 p.m. During development, the average body temperature falls slightly from infancy to puberty and again from puberty to middle age, but as soon as this stage passes, the temperature begins to rise again, and by about 18th year, it is as high in infancy. In females, during the follicular phase, which lasts from the first day of menstruation cycle until the day of ovulation, the average body temperature ranges from 36.45 to 36.7 degrees Celsius. Within 24 hours of ovulation, an elevation in temperature about 0.15 to 0.45 degree occurs due to increased metabolic rate caused by sharp increase in levels of progesterone. During fever, an elevation in core body temperature occurs caused by circulating pyrogens produced by immune system under regulation of hypothalamus.